Hello, this is Luis Enrique Coronas, and I'm a PhD student at the University of Barcelona. I'm working in the group of Giancarlo Franzese, and Giancarlo and I am presenting a poster entitled Water Contribution to the Protein Folding and its Relevance in Protein Design and Protein Aggregation. This work wouldn't have been possible without Valentino Bianco and Ivan Kolutza, and we also acknowledge Christoph Del Lago for helpful discussions. First, we introduce this topic by recalling that all atom simulations have a huge computational cost, and they aren't able to reproduce the required length and time scales that are relevant for biophysical and biochemical processes. For example, those involving proteins and water, and proteins and water and nanoparticles. So to this aim, we introduce a coarse-grained model with 3 desiderata. First, we wanted, of course, to reproduce all these large size and large time simulations within a reasonable computational cost. Second, we want it to have tunable parameters, so we can play with them, and it will be easier for us to show which is the leading mechanism behind all of the phenomena we observe. And third, we wanted to include explicit water, and we want this because we will show that local density fluctuations of water and hydrogen bond network properties at the vicinity of the proteins are essential to reproduce all of the phenomena we observe. So first, here I present the three-dimensional coarse-grained Franzese-Water model. Here, first we coarse-grain the positions of the molecules. So we keep the average distance between the molecules and not its exact position in the system. And the Hamiltonian has 3 terms. The first of them is the van der Waals interaction, in which we equilibrate the average distance according to temperature and pressure conditions. And we have 2 terms for the hydrogen bond interaction. We separate a 2-body term, which accounts for the covalent contribution to the hydrogen bond interaction, and a many-body term, which represents the cooperative effect at extreme low temperatures. We add a small amount of volume anytime a hydrogen bond is formed, so we obtain a local density field. And as we will see, these fluctuations of local density are relevant, will be relevant for processes involving proteins. We perform extremely efficient parallel cluster Monte Carlo simulations in the GPU, with speeding factors of the order of 100 or 1,000 times faster than sequential metropolis in the CPU. We are able to achieve simulation of size, including millions of water molecules and around simulation times of around 100 of seconds. Here we have the phase diagram of the model. We have open symbols representing numerical data obtained within the model, and filled symbols that represent experimental data. Here we highlight the region of biological interest, in which all of the phenomena involving proteins occur. And here we compare results, and I'm still working on tuning the parameters. So this, our quantitative, our estimations within the model are best reproduce this experimental data. In any case, we see that up to the moment we have a good agreement within the error bars. We are also able to explore the region far beyond the experimental limit at extreme low temperatures for a wide range of pressures. I'm working here on analyzing the liquid to liquid phase transition and trying to locate the so-called second critical point of water. But this work is beyond the scope of this poster, in which is focused on the results with proteins. I'm also working on extending this model with 3 dimensions with water to include also proteins. But up to the moment, we have already obtained a wide variety of results involving proteins and water in 2 dimensions. This work was performed by Valentino Bianco during the last years and many co-workers. Here, as a first example, we have the folded to unfolded transition. In theory, it is predicted that the stability region has an elliptical shape in the pressure temperature phase diagram. And this theoretical hypothesis has been confirmed in experiments, as we see here, but can't be properly seen with all atoms simulations due to its huge computational cost. So here with our coarse-grained model, we can observe this elliptical shape on the phase diagram. And we are also able to rationalize which is the mechanism underlying all of the unfolding processes. First, for high temperature, we have that the relevant mechanism is entropy. And for cold denaturation, the leading mechanism is energy. At high pressure, we have a density-driven transition, and for depressurizing, we have an enthalpy-driven transition. We rationalize all of these phenomena, thanks to the hydrogen bond properties, depending on how they vary depending on the thermodynamic conditions. So water is essential to see or to observe this the unfolding protein unfolding. Bianco and co-workers also studied protein design, in which they find a positive correlation between the temperature of design and the stability region, in the sense that the wider the temperature of design, so the higher the temperature of design, the wider the stability region in pressure and temperature is. They also studied the effects of crowding, so how the protein concentration can induce unfolding and aggregate. And they observed that starting with a folded configuration for low concentration, as it increases, the proteins first unfold and then aggregate. So again, water is here essential to explain this phenomena. And also we have studies on the effects of hydrophobic, the presence of a hydrophobic wall, which increases the protein concentration thresholds for unfolding and aggregate, and decreasing the hydrophobic effect, which is equivalent to adding salt to the solution, results in a decrease of concentration thresholds for unfolding and aggregation. So all of these studies are explained in more detail in our poster, that Giancarlo and I are looking forward to discuss with anyone interested on Thursday the 24th at room 3 during the poster session. So see you soon, and thank you for watching. Bye.